Welcome to part 2 of chapter 3 video. So back looking at diffusion. So if you take a look at the figure on the left, you're going to see, if you wanted to count them up, there's a whole lot more of, I'm going to call them scent molecules. It's actually just water on the right hand side of that plasma membrane compared to the number of water molecules inside the membrane. And then you see there are some little purple squares, and you can count them up on the outside of the membrane and the inside of the membrane. But what you see is that over time, they are going to move in random directions until there's an equal amount of water and particles on both sides of, in this case, the plasma membrane. So now equilibrium is maintained. Okay, so let's move from diffusion to osmosis. So when we think of osmosis, we're thinking about water. Okay, so diffusion of water specifically from high to low concentration across a membrane, and it's driven by osmotic pressure. So that sounds a little odd. Osmotic pressure is determined by solutes. Okay, so the movement of water is going to be dictated by how many solutes are inside the cell and outside the cell. Now, from personal experience, this is super important to know, for example, if you're going into nursing, because there are different kinds of fluids we can give people. Sometimes we're trying to draw fluids off the body. Sometimes we're trying to draw fluids out of the cell. Sometimes we're trying to get the cells to take up more fluids. So this principle of osmosis and the idea of osmotic pressure is super important to these concepts. Okay, so when we think about normal body conditions, we think about our body fluids being isotonic. Iso means same, okay? Isotonic. The concentration inside my cells is no different than directly outside of my cells, okay? So my bodily fluid that's in what we call the interstitial areas between cells is the same concentration of solutes as inside the cell. Iso. Everything's happy, right? Okay, hypotonic. So think about the solutes. Okay, this is what is going to give us this osmotic pressure. Hypo means below, so it means fewer. So hypotonic solutions have fewer solutes compared to another solution. Okay, so just remember that for a minute. Hypertonic means that the solution has more solutes compared to another solution. So generally, we're thinking about it in comparison. Okay. Now, let's take a look at the bullet here under hypotonic. So, if I were to give a patient not normal saline, but something that was lower amount of salt. So there is instead of 0.9, a 0.45. Okay, so 0.45 solution of salt. So let's think of what's going to happen. That lower than normal saline solution is going to be given to the person, and it's going to start moving through the body, and it's going to start bathing the cells. This solution has less solute and more water, okay, than the actual cell. The cell is going to have more solutes and less water. So water is going to move from outside the cell into the cell, and it's going to cause your normal cell, which was isotonic, now to swell up with more and more and more water until it bursts. Now, think about where we've given someone a higher concentration of saline, not the normal situation, but there's times to do that, usually critical care conditions. So now we've given a patient a hypertonic solution, and so there is a lot more solute compared to water outside the cells. Inside the cell, of course, a lower concentration. So what do you think is going to happen? Water is going to move from the highest concentration to the lowest. So the water inside the cell moves out. It causes our cells to shrink, and it's called crenation. So these are concepts that you're going to want to understand. There is a lab on this as well. But essentially, when we think about tonicity, we're thinking about hypotonic, isotonic, and hypertonic, and how that might affect a blood cell. So it's super important in most conditions to maintain homeostasis, and in healthy bodies, we are thinking about our cells being bathed in isotonic solutions, which is 0.9% normal saline. Okay, facilitated transport. Now, when I see facilitated, that tells me, oh, you need some help. So these are movement of molecules across a membrane from high to low concentration, but you need a protein carrier or a transporter, okay? So essentially we have to work a little harder to get it in, but it needs to go in. Now, it could be passive, so no energy required, but there are certainly protein transporters that do, in fact, need energy, so that's a whole nother thing. But for our protein transporters, we know that they're very specific and they're only going to move certain things. So like a glucose transporter, as its name implies, only transports glucose. Okay, it's not going to transfer lipids. It's not going to transfer any kind of amino acids or anything like hormones. It's only a glucose transporter. Okay, so here we see glucose represented by its stoichiochemistry, I think is what they call that. So here we have it, and we have this special glucose transporter, and it is bringing that across the membrane, facilitating it. All right, so let's take a look at active transport. So active transport makes it sound like, okay, facilitated, you needed help, but didn't need energy. Active transport, you need help, but you need energy. But look here, active transport is different from passive transport because we're going to move molecules from lower to a higher concentration. Everything else we've talked about goes from high to low, diffusion, osmosis, tonicity stuff. And then lastly, facilitated transport. Those were all going from high to low. So active transport means we are going against the concentration gradient, so we have to use some ATP. ATP is the version of energy that cells use. Okay, my car uses gasoline or it uses electricity. My cells use glucose, but it's going to make ATP. So ATP is the version of energy that cells are using. We consume glucose or other energy-containing compounds or similars so that we can make that ATP. Okay, so to do active transport, we need energy. We're going to go from low to high, and we need a pump, right? So we need a special thing that's going to be doing this. It's not just going to like passively move across the plasma membrane. And the really excellent example of this is a sodium-potassium pump. So we use sodium-potassium pumps for action potentials in the nervous system. So sending those signals along the axon membrane, we're going to use sodium-potassium pumps. So essentially, to force it to do that, we're going to use some energy, and we are typically going to be moving sodium ions out and potassium in, so active transport. So here we see, there's the ATP we need. If you're curious about ATP, triphosphate, DP, diphosphate, we rip off a phosphate, and that's actually what's generating the energy. It's a high energy phosphate bond, so it generates like a spark, if you will. But when we do that, we leave this poor little ADP, kind of denuded here, and it can become ATP again when that high energy phosphate bond is put on. So you see here, we are pumping sodium out and potassium in, and so we are doing that through this special protein. Okay, protein pump, sodium-potassium pump. Okay, bulk transport. So this is super cool, a really great, easy way to move large molecules across the membrane. I mentioned it earlier a bit. Endocytosis, we're taking in either big things into our cell. I'm an amoeba. I'm eating a bacterium. Yum. Okay, and so that's phagocytosis. We also do that with our white blood cells. Our white blood cells can phagocytize bacteria and essentially eliminating them from our bloodstream, which is super cool. Pinocytosis generally means it's the endocytosis of little tiny things, so small particles. Exocytosis, my favorite example is contractile vacuoles inside microorganisms like amoeba. They got a lot of waste products. They essentially have to dump it, no pun intended. But essentially this happens with this vesicle, this membrane-bound structure that has all the waste in it. It's going to go to the plasma membrane and it's going to exit the plasma membrane. When membrane fuses with membrane, it dumps out, or when membrane fuses with membrane, it can bring it in. So that's essentially how bulk transport is occurring. So here's a cute little figure, big things, phagocytosis, little things, pinocytosis, and then the last one just means, hey, receptor protein or receptor mediated means the cell is willing to invest a little bit more of its resources to get it right. All right, let's take a look at the endomembrane system. So essentially what we have inside our eukaryotic cells is a series of membranous organelles that function to process materials for the cell. It's like a whole manufacturing center, which is super cool. And it starts kind of as its central focus, the nucleus. Okay, so let's take a look. So here we have the nucleus. You see the nucleus has nuclear pores. This means things can exit this nucleus. Like the messenger RNA can exit the nucleus, and then it's going to be intercepted by this membranous set of chambers, or channels, I should say, called the rough endoplasmic reticulum. It's rough because it has ribosomes on it. These ribosomes are important for protein synthesis. So I'm sending out some messenger RNA. It's going to go right here, and it's going to start to make proteins, which is then going to go to the smooth ER. We can manufacture some lipids and other things there, and eventually it goes to the Golgi. Hold on. The Golgi apparatus, which is going to package and label those materials and tell them where to go. And then finally, we are going to take membranes again and surround these materials to tell them where to go. So the Golgi apparatus bags it and tags it, and then essentially it tells it where to go. Okay, so this is the endomembrane system. You got the nuclear envelope of the nucleus, the endoplasmic reticulum, the Golgi apparatus, and vesicles. Lysosomes contain digestive enzymes and are there to degrade away waste products. Okay, so if you want to pause here and take a look at some of these terms, you can also find a bit more detail in your textbook about them. So essentially the rough ER, as I mentioned, is going to be this endomembrane system, has some channels, and it's studded or has on it some ribosomes, and that's important to making proteins. Then you're going to see that outside that rough ER, you have the smooth ER, so it does not have the ribosomes, so no studded features. And mainly what we're doing there is synthesizing lipids, but there can be some variation. And then those materials that are being made from the rough ER and the smooth ER go to the Golgi, our packaging and labeling center. So essentially, it's going to make sure it's ready for transport and puts tags on it. And essentially those tags are going to tell those materials where to go. We package our materials in vesicles because why not? Instead of using envelopes like you might if you're sending mail, we're going to use membranes because it's kind of like this universal chemical of life. And so it's really super easy to put things in vesicles. So if I'm going to do exocytosis, my vesicle with this membrane goes and fuses with the membrane of my cell. So pretty handy. And then you kind of just reuse those the membranes in the plasma membrane, the cell membrane. Lysosomes, so as I mentioned quickly a minute ago, they contain enzymes that are going to break down molecules into smaller parts. So be thinking, we build up wastes inside living things. And so lysosomes are one way to eliminate some wastes. Lysosomes are particularly important in white blood cells because they can engulf disease-causing microorganisms. So another example here of white blood cells having a critical role in taking care of waste. Finishing up here, the cytoskeleton essentially is a network of protein fibers in the inside of the cell, and it's going to give that cell shape. It's going to anchor some organelles and let other organelles move around a bit in the cell. So think about it. You've got a skeleton. I mean, most of life has some kind of skeleton inside or outside, so, reinforcement. 3 types of fibers here you might learn more about in another class if you go on and take a PAP class, Principles of A&P. But essentially we have microtubules, the largest of these elements that make up the cytoskeleton. We have some medium-sized, and then we have actin, which is really important to movement. Okay, I'll see you next time.